Testing. Mic to earth. Okay. Welcome, everybody, to the next installment in the Fralin Biomedical Research Institute Pioneers in Biomedical Research Seminar Series. That's all I'm going to say because I'm turning over the introduction to Dr. D. Felice Antonio, who will introduce today's speaker. Alex.
Thanks, Mike. I'm really excited to introduce Dr. Kyle Berger today as today's Pioneer in Biomedical Research Seminar Series speaker. A bit about Kyle. He did his bachelor's in nutrition and dietetics at the University of Nevada. And from there, he went on to get a master's in public health at San Diego State and then did his dietetics internship at Tulane. So he's just racking up the letters after his name. And then he went on to work with Sue Johnson at Colorado State University from his Ph.D. From there, he did his postdoc with Eric Stice at the Oregon Research Institute. And he was really prolific during that time, publishing many papers, including one of my favorites that I cite often that appears on PubMed when you use the search terms ice cream burger. So if you want to do that, you can find that paper. In 2013, he became an assistant professor at the University of North Carolina in Chapel Hill, where he started the Neuropsychology of Ingestive Behavior Laboratory, or NIBBLE, which is a great name, but also, Kyle, you have the best last name for someone as an ingestive behavior researcher. And if you were just going to go with last name lab, it would have been a good thing. And so, Kyle has been extremely prolific, publishing many papers on the neuroimaging of food reward and eating behavior, also how it intersects with development, which I know is of interest to a lot of people here. He's had ongoing support from the NIDDK as well as NICHD, and I've really admired his work for a long time. I'm very much, if you want to do something right, you should do it yourself. But if you want something done right in neuroimaging and nutrition, you can also hand it to Kyle. And so I'm really excited to hear about the new data that he'll be presenting. I'm heard it's brand new about the cyclical nature of food reinforcement, decision-making, and overeating. So thank you. Please welcome Kyle.
Thanks. Thanks so much, Alex. That's quite sweet and not all deserved, but so I am really excited today. Sometimes you go to talks and people are like, oh, we have brand new data, and you're in the audience and they're like, that's great. I don't know if it's new or old data, but this is like my new data that's been like 10 years in the work, 15 years in the work. So we'll get to it. I'll set you up there. But first, just to give you a little roadmap, we're going to talk about obesity-related brain functioning at rest briefly, and then kind of what it looks like to evoked response, meaning to food stimuli, whether that be pictures, anticipatory cues, or receipt. Then I'll briefly touch on some of the prominent food reward-related theories of obesity and where the gaps in knowledge are and the assumptions made. And that leads us into talking more about eating behavior and brain response, eating behavior, decision-making, habit formation. Then we get into my new data, which I use the causal word there, which still I'm not comfortable with yet, but still. We'll talk about sugar causing some neurobehavioral adaptations that are similar to substance abuse, and then briefly discuss kind of our framework to understand that. First, just looking at BMI and resting state functional connectivity, we did a study using the Human Connectome Project data in 47 pairs of twins that were BMI discordant. And so you'd think that, they're twins, so they might not be that different in BMI, but when you look at the most discordant twins, it's about 6.5 BMI units. So that's a full BMI category. So that's quite a bit. Obviously, they're adults, and so it's not like the twins are exposed to the same environment at that time, but they are similar in genetics. And what we found there is we found stronger connectivity in the higher BMI twin with insulin, oral somatosensory, and anterior cortex with this right crest of the cerebellum. So what that means is, or one interpretation of that is, this stronger cerebellar insular connectivity is associated with caloric deprivation and hunger, previously associated. So in high BMI individuals, stronger connectivity of these regions might be associated with compromised satiation signaling or increase in duration of consumption or postprandial meal food intake. Looking at the other side, we saw stronger connectivity in the lower BMI twin in the striatum and the thalamus with the dorsolateral prefrontal cortex. And of course, the striatum is known to be a dopaminergic region associated with hedonically motivated eating behavior. And the dorsolateral prefrontal cortex is, of course, very similar, very well known for top-down executive functioning, cognitive control, those kind of things. So this could indicate the stronger connectivity in the lower BMI twin could increase regulation and decision-making when presented with highly palatable foods or foods in general. Looking at kind of a review article, just to kind of keep things moving, higher a recent review article, similar effects you'd see. Higher BMI is associated with increased activity within the salience network, increased connectivity among the striatum, posterior mid-insula, posterior cingulate, and somatosensory regions. So, striatum, hedonically motivated behavior. Posterior mid-insula is gustatory processing, but also, more importantly, probably craving. You see that with smoking and alcohol as well. Posterior cingulate, salience independent of valence, so attention and salience processes, and then somatosensory region. Specifically, will you see that as the oral somatosensory region? So mouthfeel, those kinds of things. So what about brain response to food stimuli? And here I'm talking food cues and food receipt. And so this is my stick figure assistant named Stanley. You back him into the fMRI, you have a mirror, you're looking at whatever food stimuli it is. In that case, it's a bottle of juice with our fake little logo on it. You have pumps externally that will pump liquid into that. The paradigms are set up. The paradigms that are set up is they see a picture. There's typically a weight, they receive a taste, whether it be a milkshake or a juice or those kind of things. There's a break, a swallow cue to control for head motion, and a rinse, a tasteless rinse. Also you do, because you need an active contrast, because you don't want brain response to just something in your mouth. You want brain response to, let's say, like Alex says, a milkshake. You want brain response to this highly caloric, highly sugar, highly food, not just liquid in your mouth. So you have a contrast of a taste solution or flat water. So when you do the analyses, you essentially subtract out brain response to the water or tasteless solution to get to the palatable part of the milkshake or whatever the active part of the contrast is. This is what it looks like in real life. Those are my sexy forearms on 60 Minutes. And this is me drowning Sanjay Gupta in Coke. I do not recommend putting a GoPro in an fMRI. Just throwing that out there. All right, so what's the neural encoding of food stimuli? There's been a number of meta-analyses at these points, so just focusing on those. Again, you'll see the similar brain regions that we saw with resting state, where brain regions that are associated with hedonically motivated behavior, craving, gustatory processing, and oral somatosensory. You see that commonly, actually remarkably commonly, as I was looking into the meta-analyses, as more and more of those are coming out. And what you see is these regions are typically heightened in those individuals with obesity when they're presented with food cues. Now that last part is important because we're looking at food cues relative to the intake of food, which we'll see why it's important here. So this first theory that I talk about that you'll hear is a hyperpalatable foods or food cues are causing obesity. So it's this immense rewarding aspect of these highly processed, high sugar foods that perpetuate intake. And, you're attributing that to the food cues, you see it, so you just got to have it. This is a pretty prominent theory. However, this reward related thing. However, reward's far more complex than that. I always feel like that's kind of like a pop culture explanation or pop science explanation of it. For example, the other theory is a blunted striatal response to food intake or just a blunted striatal response in general. Because with obesity, you see decreased dopaminergic functioning or dopamine receptor binding or dopamine receptor density in individuals with obesity. And then also when you see individuals with obesity respond to taste of their mouth, so the actual consumption of the food, you see a decrease in response in these regions as well. So at this point, if someone in here studies addiction or knows this literature well, you say, Kyle, these are models of substance abuse or substance addiction. And they really are similar and they're applied to that, applied to this kind of food thing. And I know that's we'll get into the concept of food addiction a little bit later on, but they are similar in the patterns that you see. But like I said, reward isn't really reward, at least it's not all dopamine. I think that's a big thing that we lose when we're talking about this. So for example, the incentive sensitization theory by Kent Beverage and Terry Robinson, and then more recently, the dual pathway model by Dana Small and Avante Rojo, suggests that perceptual liking, so that doesn't impact caloric intake, and it isn't encoded by dopaminergic response, really. So it would be more, and this is where you follow this liking, and where liking describes the perceptual hedonic impact of a food reward. And that doesn't necessarily mean that it is perpetuating food intake, whereas wanting, referring to the incentive salience or a cue triggered motivation, for example, a craving or hedonically motivated behavior, is what's driving future intake. Yes.
Sorry, the positive theories that if somebody is obese is not getting the satisfaction of the reward from the food, but there's got to be some reward in the behavior somewhere. Is it delayed in that I'm hungry? There's a hunger signal, and reward is the hunger signal is satisfied, but I'm not getting reward while I'm eating or seeing the cues. Is that?
Yes. So that's kind of the Nora Volkoff thing, and you go into George Koob too, which is the reward alleviating hunger, which is an uncomfortable feeling like that, or are you so it's almost like a relapse model versus are you not getting this dopaminergic response while you're eating, so you're perpetuating trying to catch up. It's like, oh, if I eat more, I'm going to get more of that dopaminergic response. Yeah.
The thing that's missing here, though, is that eating is a very systemically different thing from taking, including that it engages in a whole other part of the nervous system. It's bigger than your spinal cord, and that everybody does this sort of thing, seems to an artificial way, which is the enteric nervous system and its interface with the immune system. Mm-hmm. And so it seems to me that in thinking about these things, you really need to integrate those. And so I'm wondering how you think about this, even in this model, because it seems like it ignores a key aspect of it.
It does. And Alex is doing a lot of this work. As this has evolved, because this is relatively, on the hierarchy of things, this is relatively new, like brain imaging and food, 10, 15 years or more than that now, I'm old. But you're moving towards the interaction of the gut-brain axis as well as inflammatory kind of responses and those kind of,
to really have an effect on the entire way the brain is operating, being that it's an organ. So
and actually in the dual pathway model, which is I'm more than happy to share it with you, but it's a paper presented first in a paper called Rethinking Food Reward. That was the first one to really bring in gut-brain access into this, and it's conscious liking versus subconscious wanting, and the reinforcing signals coming from the gut. And then of course there's variability on macronutrient content as well. So how does this wanting land with predicting overweight or obesity? There's a meta-analysis done by a colleague of ours. This is just subjective wanting via questionnaires or visual analog scales by our colleague Hedy Kober. Cue potentiated food cravings, so wanting in response to food cues can predict eating as long as long-term weight outcomes. Tonic cravings, which is just more asking in general, do you crave chocolate? Those kind of things, not cue potentiated. Also predicts predicts increases in eating and then future weight gain. So here we have these kind of 2 or 3 models of obesity and food reward. You have the striatal response to the food cues, images, wantings, the cravings. You have the decreased striatal functioning, which is that possibly compensatory action or relapse model. You also have, which I didn't discuss much here, is compromised prefrontal response, because I think those kind of fall into the above, but more poor cognitive control when faced with food stimuli. So what my first question was when I was doing my group is these are all theories and models of obesity. They're tested in obese versus lean individuals. Some of them, only a handful of them, are using prospective work, but they're also using individuals at a wide BMI range to predict future weight gain, when we know that high BMI is the biggest predictor of weight gain in the future. So do these, what about the impact of altered neuroendocrine functioning when there's associated with excess adipose tissue? Like, is this just a maintenance factor? Is this just, how does this initially unfold? Because prevention is more important than treatment if you can swing that. So do these things, are they a function of behavior? So the first thing that I was looking at is I was trying to figure out how wanting arrives, this wanting of the cue. Like, where does this come from? And I started looking at reinforcement learning and decision-making control systems, which we'll circle to in the back end. But first, we just wanted to look at Pavlovian conditioning. Very simple, very, very simple. Like I said, we'll talk about habit-driven and goal-directed control later. But the first paper, when you're thinking about sensitization of cues, is you might think of Wolfram-Schultz work, where in an unconditioned portion, when a light preceding a taste of juice, I think in this case, before the light is paired with the juice, the dopaminergic response occurs to the juice. And then after pairing, the dopaminergic response occurs to the cue and not to the juice, suggesting that the cue is sensitized and has like that motivational target, as Kent Bird would say. So I was trying to mimic this in humans. And so I took, I think, as secondary data analysis of a number of people, and an MRI of multiple runs, multiple exposures. And so I just kind of chunked them off and then looked at the change in striatal response over time to a food cue that was just a geometric shape. So basically what we saw was, as the people were pairing the shape to this chocolate milkshake, just through simple passive exposure, we saw an increase in caudate response over time. So it falls directly in line with what Wolfram-Schultz work was, and as far as the insensitive sensitization kind of model, that the cue is becoming more salient. Interestingly enough, we did see a decrease in ventral striatum response to the taste over the same amount of time. So that's somewhat akin to this hypo reward or model that way. Perhaps more interestingly, we did see that the rate at which the caudate response was paired with the cue, so that sensitization of the cue predicted future weight gain 2 years out, I think. Yeah, 2 years out. So that was pretty promising. That was nice. Nice use of secondary data. And so my second question was, can we see these patterns that are obesity, this hyper response to the cues, blunted response to taste? Can we see that as a function of eating behavior, not weight? And so we looked at doubly labeled water estimates of caloric intake over a 2-week period in lean adolescents, and then assessed how many calories they were consuming over brain response to a milkshake cue. And in this case, we saw greater response in the precuneus and the frontal operculum, anterior and visual cortices. And so that's very similar to that model of obesity. But remember, this is just a function of objectively measured overeating or eating beyond basal metabolic needs. But it is the similar pattern to the heightened response to food cues, ads, anticipation, wanting, and craving. So looking at the flip side of that, this is your brain on milkshake, and circled there as a striatum. Really, really strong response to milkshake relative to the tasteless tasteless solution receipt. When you regress on how much people are consuming frozen dairy desserts like milkshakes over the previous 2 weeks, you see it completely flips. So the more an individual consumes ice cream or frozen dairy dessert type thing, this was assessed via questionnaire. So take that for what that be. This is the burger ice cream study that Alex is talking about, if you're interested. But yeah. So the more individual consumed ice cream, the less striatal response they'd have. One of the things that I overlooked in that paper is, there's a lot of, in the striatum, there's a lot of encoding of novelty. So it doesn't necessarily mean this is this huge behavioral impact. It just could be novelty. So I moved on and I wanted to look at sugar sweetened beverages is always an easy target for us nutrition folks. So I took habitual Coke consumers versus non-consumers. So they had to like Coke, they didn't drink anything else. And then the non-consumers didn't drink any sugar sweetened beverages. And so I showed them pictures of the brand of the Coke and then pictures of non-food brands. And then I showed them pictures of the stimuli, the actual product as well. So greater response to the brand versus a non-food brand. So just like, I don't have pictures right here, but just like the Coca-Cola logo, not anything that showed the product. So we saw greater response in the Coca-Cola consumers in the precanous again, or posterior cingulate, if you want to go that way. However, when you showed pictures of the images of the actual product shown there was no difference, suggesting that the brand had a little bit more salience to individuals that were consuming. But when you're showing the actual product, it's just kind of everyone knows it's Coke. So it's like, cool, not that big of a deal. So next step, as we kept moving forward, and again, this is independent of BMI, we did a quasi experiment. So it's a randomized controlled trial to assess the impact of daily consumption of a branded sugary juice. We just mixed juiced flavors together to make it novel over a 3-week period. Now we only had 20 people in this one, 10 males, 10 females. These people were BMI within a recommended limit range by design. We didn't want that confound of excess adipose tissue. They did not consume sugar sweetened beverage or sweetened beverages or artificially sweetened beverages. So it was a pretty non-generalizable but clean sample. Small sample size, no pure control. In this case, they were randomly assigned to consume one of the 2 sugar sweetened beverages, two juices, and they were tested always on the consumed sugary juice versus a non-consumed sugary juice. So we can't make inferences about sugar because we have no sugar control. We did a behavioral assessment, which I'll go to in a second. We did an fMRI, which I'm not going to present the data on today. They did 21 days of juice consumption. They came into my lab. They drank it in front of us. They got the bottle for tomorrow. They came into our lab the following day, or like on the Wednesday, brought us back the bottle for adherence, drank the thing. So we at least know that we at least saw them consuming it 9 times. So at minimum. Cool. So some of the assessments we did this version, it was a go, no go task. So we were looking at motor disinhibition towards the juice logos of the consumed versus the non-consumed. And again, they're just responding as quickly as they can and then inhibit the response when they're supposed to. We did this at pre and post intervention. We did visual analog scales, -100 to 100 for liking or pleasantness of the beverages and desire to consume, wanting at pre and post as well. And they did that for their consumed beverage and non-consumed beverage. So what we found was a significant time by assignment interaction. So faster reaction times were recorded when they were responding to their assigned juice, and slower reaction times were recorded to when they were responding to their unassigned juice. So again, so that would suggesting there's maybe a greater implicit approach behavior towards their consumed brand. And when I say responding to the juice, I should say the logo, their brand, which is just like that basic black and white logo. Cool. The other thing that we found was in looking at the visual analog scales is we found that there was always a relative decrease in pleasantness and desire for the non-assigned juice. So you're consuming whatever flavor in this case, you begin to like the non-consumed beverage more, or you like it less. So one of the ways I think about it is like a Pepsi Coke kind of thing. Like a lot of the times you'll ask people, which you like better, and they'll probably say Coke based on the national average, but people say, well, do you really like Coke? Not putting it in that context. And they'll be like, oh yeah, I like Coke, but they like Pepsi less. That's what's driving them towards Coke, is not that they love Coke so much as they don't like it relative to Pepsi. So this again is just kind of driving them towards towards their consumed beverage. So yeah, sorry.
If you sort of vary another aspect of the stimulus, like the color, but the taste would be the same or only moderately different, but you could make a discrimination. Would that be a control? Or the response is to the logo, not to the juice.
Yeah, I see what you're saying. So are you suggesting making the logos more distinct, or no, just
the juice stimuli different on another dimension?
We will here in a minute, but we do have a pure control. But
blue juice versus red juice, or green juice versus yellow, or something.
Yeah.
Because there's a visual component to the discrimination.
Yeah, no, exactly. And we're really close to answering that one for you. No, but yeah, it's a great point. So here we can just say, these patterns we see people that are very into food addiction says, this is food addiction. And we simply induced a habit. We experimentally made people have a habit. We can't draw inferences about sugar. We can draw the inference about relative consumption of one beverage to another. We can say that we did induce this habit, and they're neurobehavioral patterns that mimic, in theory, that would influence food choice and habit formation. But this again is a small sample, no control group. And we don't know if this would occur with just like a colored water, or a flavored water. And we don't know if it would occur with some sort of healthy kind of rewarding activity like physical activity. So then we move on to the COVID trial. We had a randomized control trial. So this one's actually, we have a control. And this one, we actually have 2 flavors nested within each condition. However, I did not analyze that. So this is a brand new data. We were at 160 people that are assigned to consume. I'll be presenting data on 122 that have valid pre and post fMRI data. So again, it's a very similar pattern. fMRI, we create these little logos and brands. We have the visual analog ratings, and then we also do a reaction time test here again. Again, they come into our lab 3 times a week. They drink it in front of us. We get some ratings. We send bottles home every day or 2 on the weekends. They bring them back for compliance. We have currently 5 pre-registrations on this now. Yeah.
The unsweet one in this, without a non-nutritive sweetener, or just like unsweet?
Yeah, it's just flavored water. No sweetener, no sugar. It's hard to kind of, no sweet, no sugar, just the flavor. We were originally supposed to have 250. We were cut short during COVID. COVID messed with your taste. Couldn't really bring it back. We had planned a washout phase to where they would come in for a scan and behavioral assessment roughly 10 days after they went through the intervention to see kind of if the effect was sticky. That phase was set to start the day our campus shut down. And like I said, I'll present completers analysis essentially on the 122 that have good data. So the branding, the active condition is 111 calories, 27 grams of sugar. That's equivalent to like a soft drink. Unsweetened control, no calories, no sweeteners, but they're the same flavor. We went through pilot testing that I won't go through about determining the flavors and those kind of things, but and like pleasantness of the logos and those kind of things to make sure try. So the analytic sample, we had 75 female. They were equally, well, that's the majority. They were distributed equally among the assignment to sugar and assignment to control. Our average BMI was slightly higher than the previous trial, but not much. But we did have individuals that were presented with obesity and overweight. So looking first at the baseline correlates, neurocorrelates of liking and wanting to the sugar brand. So this is during the sugar cue exposure. And what we saw during the sugar brand exposure, wanting greater than liking correlated with central operculum, visual cortex, dorsal ACC, anterior cingulate, and fusiform gyrus and somatosensory gyrus. Again, these are pretty common regions. The interesting thing here is this is that, we're thinking about that wanting, that desire, that craving, that sensitization of the cues. you'd expect to see nothing with the liking. Well, we didn't see anything with the liking. So that kind of falls in line with that, despite them being like subjective. And I think the idea, kind of back to what you're saying, is the idea that wanting is a little bit operating more on a little bit more of an unconscious level, an unconscious drive relative to this explicit, I want this. Wanting is notoriously hard to measure in humans, and liking is notoriously hard to measure in rats, in my opinion, but there you are. So let's look at the impact of daily sugar consumption. This is kind of the main finding. What did we see? So because we had a larger BMI range in this, we know that there's aberrant response in individuals with obesity when presented with food cues. We first tested a BMI by time by assignment interactions. So the effect of the intervention, does it vary by BMI? And so what we did see was sugar consumption resulted in a greater mid-insular response putamen and central operculum to the sugar brands. And this was higher in those with higher BMI. So digest that a little bit, sugar causing altered brain response to the brand. And this is particularly strong in individuals with higher BMI. So immediately what you're probably thinking is like, well, this doesn't, okay, so this isn't that profound because we know that these patterns exist with individuals with higher BMI. But remember, this is versus baseline. This is a novel taste, novel cue. We're creating this response. And moreover, in a post hoc test, I just took the sugar sweetened beverage group and divided it by BMI categories. And above 25, which would be classified as overweight or obese, the effect was still there. So this suggests that the result is generalizable to 70% of the population. So yeah. I don't know.
If there's a taste drive part of this,
Yeah,
because that's where the taste cortex is.
Yeah.
One assumes when you're taught. So that's a surprising result.
I don't know why it's weaker. I can tell you that it's bilateral when we're sub-threshold, and we use a pretty stringent correction for multiple comparisons. If you use a cluster-based correction, it's bilateral. And that's actually most of the case with all the findings we have. So there's some other, depending, if you do brain imaging analyses, you know that you can get away with a lot, and some people do. But I think we try and lean really hard on losing false negatives as opposed to presenting false positives. But it is bilateral, and when you drop the threshold a touch.
One more question.
Yeah.
You mentioned the difficulty with measuring wanting and liking, some challenges associated with that.
Yeah.
How did you measure it in this set?
Oh, this is just a visual analog scale. And that's why I was actually surprised to see, because liking and wanting are highly correlated in humans. The time that you can see individuals consistently differentiating between liking and wanting is when you do a comparison of individuals with obesity and without obesity in a fed and fasted state. And typically you'd see individuals within a healthy BMI or a recommended BMI range, you'd see liking and wanting be really similar after feeding. Liking would be the same, wanting would drop. The wanting would not attenuate in individuals with obesity. But here, that's why I was kind of surprised by it, and I kind of put it in. And the fact that the regions seem to be falling in line so consistently with even the subjective wanting at baseline. I haven't done the subjective wanting or the interaction over the intervention yet to see if that changes. But alternative methods for assessing wanting that are behavioral, which I'm a huge fan of. You have like progressive ratio tasks, you have those kind of things. They all have their drawbacks. The closest thing, and we're doing a lot of eye tracking now to try and, because what I really want to measure is implicit wanting. And like you have either like perceptual explicit stuff or explicit stuff, pressing the button to annoy the RAs forever. But yeah, right now we're doing eye tracking and essentially like a human-sized operant box and seeing if we can get stuff out of that. We're aiming for sign tracking, goal tracking, if you're familiar with this. But so what does this look like? What do these regions look like in context to some of the previous work? Would this pattern, does it exist anywhere else that you've seen? And you look at my old postdoc's boss in a paper, and these are the 3 regions that predicted weight gain over 4 years follow-up in response to a milkshake cue. To be fair, I figured that out last night. So I'm just kind of like, it seems too similar to me. So that it's different sample, different part of the country, different everything. But I just kind of found that fascinating. So moving on to the behavioral. So we have, again, the reaction time test. And so here you can see that there was a non-significant interaction between the between the assignment and baseline follow-up. However, when you're looking at individual change within group, you do see a decrease in reaction time in the sugar sweetened beverage group to their things. So that looks similar, and despite the fact that it's not a significant interaction, it's the same pattern you'd see from our initial study from 5 years ago and the smaller sample size. Faster reaction time toward the logo consumed. Here we can just show that it occurs in sugar and not in a control group. So the perceptual ratings, again, now we're back to this liking and wanting. There's no difference in liking in the significant interaction, but that was driven by increase in liking of the non-sugar condition, the control condition, which the no change in subjective liking of the sugar sweetened beverage is consistent with what was previously found, but this increase in liking of the control condition is a little bit curious. And so I was looking at it, we have a measure of familiarity. It follows that directly. Basically, the sugar sweetened beverage is more familiar. This pattern's almost mimics it. And then if you think in terms of like preference development and food neophobia in kids, it takes about 8 to 12 exposures before you start developing preferences. And I think we don't have a bitterness rating, I believe, but I think like subjectively, the unsweetened beverage is a little more bitter. So over time, familiarity breeds preference. Yeah.
Minus liking. Here, did your analog scale go from?
Negative 100 to 100. Yeah. Sorry. Yeah. So as far as wanting, there's a main effect, no interaction. So both groups increase their wanting slightly. The magnitude of the effects are relatively small, but the patterns are there. This, I will say, when you're doing randomized controlled trials, a lot of times you'll do intent to treat analyses and multiple data imputation, those kind of things like this. This effect right here is, even though it's a main effect, it is driven by the unsweetened control group, similar to that guy. Yeah.
Analyze subjects. In other words, you have the baseline and follow-up, drew the line between the same person. What would this look like?
So I don't have that graph. So we actually took measures of every time we saw them, of liking and wanting of their own beverage. And for the liking in the unsweetened control group, it was at assessment 5, exposure 5 through 7 is where it ticked up, and then had just like a moderate slope. The variability, there's always a lot of variability in these, but essentially the sugar sweetened beverage group was pretty much flat. I think they tick up on the second time they taste it, and it's pretty much flat with a slight increase.
In terms of there's a group that
not that I teased out yet. But I did look at that in the previous trial a long time ago, but I just didn't have enough people. But that is definitely like, we had these spaghetti graphs, and yeah. Exactly. And that's one of the things that we're hoping to do that I haven't done yet is do that analysis where you're looking at the changes in brain response over the sensitization in the first time they're in that magnet, kind of like that Schultz thing. I wasn't able to get that done. I did it a kind of cheesy way in the effects there, but I'm not allowed to talk about it because that's a cheesy way to do it. So these are the perceptual ratings of the assigned beverage. And so now we're going to look at the perceptual ratings of the unassigned beverage. So it's a little bit, you just have to take a second to wrap your head around it. So those in the sugar group, sugar condition that consume the sugar sweetened beverage decreased their liking of the alternate unsweetened one. And those in the, those not assigned to the sugar condition, the one that were drinking the control, increase their liking of the sugar sweetened beveraging. And you see this pattern, it's actually, sorry, that was not a significant increase, but this is. So you see again this pattern that patterns, the devaluation of the non-consumed beverage is consistent with what we saw previously. Yeah, so it's kind of nice how it lined up again. Like I'm the first to say the effects are not giant, and when you're looking at like magnitude of visual analog scale, though, and I mean beyond statistical significance, they are meaningful changes and trends. And keeping in mind that we're 33%, or we're only 66% of the sample we had intended on recruiting. So in summary, we got consumption of a branded sugary beverage resulted in an increased brain responses associated with hedonically motivated behavior, craving, gustatory processing when exposed to the brand. We did see an increased behavioral approach, like speeded response to their assigned brand, the sugar in the sugar group. And then we see decreased valuation of the sugar-free is a poor term there, but the control beverage. I kind of deemed this hedonic targeting, kind of towards the sugar, just kind of the combo of those. And this is fitting into our like kind of an evolving model that we have going that is eating habit architecture, where it's just basically a reinforcement learning based model of compulsive and impulsive overeating. And so what does that look like? Well, initially it's pretty much three components based on the three decision control systems, or reinforcement learning, or decision control systems. One, you have Pavlovian decision-making control, where you have Pavlovian learning. We talked about that a little bit previously. Pavlovian decision-making control is really innate. It's very reflexive. It's very inflexible. It doesn't update to changing reinforcement schedules. It is sensitizing cues that approach, sensitizing cues creating approach behaviors. And aberrant Pavlovian control could be a mark of impulsive behavior, where this is impulsive behavior is defined as a predisposition towards rapid, unplanned reactions to external and internal stimuli independent of the consequences. The second component is goal-directed decision-making, if you're familiar with decision-making models. This decision-making model it's learned, but it's highly flexible when facing changing reinforcement schedules. So it's sensitive to the state. So in this case, this would be, in theory, have a low risk for overeating because it doesn't permanently sensitize cues. If you're eating and the evaluation of the food that you're eating changes, or you have stomach distension, you're able to update your decision-making. So this would be a lower risk. And then lastly, habit-driven choice or habit-driven decision-making. This again is similar to Pavlovian control systems, but where it's rapid, reflexive. It's highly inflexible because it doesn't respond to prediction error, which is unanticipated change in reinforcement. I think this isn't as really well fleshed out, but I think people forget that susceptibility to overeating can be in response to a food cue, frequent meal initiation, but it also can be because of delayed satiation, longer eating at a single occasion. And I think this habit-driven choice system, a propensity for that, could result in like lack of sensory specific satiety, lack of satiation processing, so they continue to eat. And this is kind of a mark of compulsive behavior, where compulsive behaviors are repetitive and habit-like actions without necessarily leading to a reward. You're just doing it because you're doing it. So here, we've shown that sugar intake caused a stronger sensitization towards the cue, started towards that brand. We did see kind of a transition from goal-directed choice to habit-based decision-making, in that it became more reflexive and automatic, though we did not measure behavioral choice directly. So, and then the question whether the individuals are insensitive to a changing reinforcement schedule, we haven't analyzed that. I didn't mention it, but we have a reversal learning paradigm at the end to see if we can unpair the sugar cue with individuals, in particular to see if the group in the sugar condition is able to unpair the taste from the cue as readily as the unsweetened condition. So these are key factors in compulsive and impulsive, like overeating, but is it food addiction? Is it food abuse? And then that's where we can just have that spirited debate about that. First, I'd like to thank my lab and Russ Boljack and Jeanette and Dana Small, as well as Alex and Carissa. Appreciate all the help, and I really thoroughly appreciate the invite to come talk. It's been lovely, and I look forward to talking to more of you soon.
Anyway, thanks. It's really interesting. I learned a lot. I'm kind of wondering at the large level about memory and where that fits in here. So I was wondering about it in three contexts. One, just in terms of the functional imaging and the networks and stuff. Two, Anthony's question earlier about these subgroups possibly coming out. And three, on the behavioral side. So what I'm getting at is, are there different people who in general, their memory functions are better or worse, but specifically in the domain of remembering reward signals from eating behavior, for which those differences could be important reinforcers over time to modulate behavior. I didn't really notice in your fMRI stuff a lot of the places I would have been looking for to see kind of memory signals.
Actually, I had a hypothesis, and hippocampus was pulled out specifically and didn't show up. So you get to be wrong a lot with a priori hypothesis. But we do know that their obesity is associated with working memory deficits. I'm not 100% sure. I can't comment really on whether that's food specific or not. Though I will say I do know, in a very simple kind of task, Ashley Gerhardt, I think she's first on that paper, they're showing kids food advertisements in the magnet, and then just do a check and non-food advertisements, and just did a checklist of what advertisements they saw in the fMRI, and individuals with obesity, or children with obesity or elevated weight, I think in this case, remembered food specifically. But the goal-directed choice does engage working memory, because it's how it's, and then there's better people with computational decision making in the room than I am, but it does involve working memory, because it is frequently reevaluating everything based on the changing infrastructure. That habit-based does not engage working memory at all. It's just too reflexive for that. But it's a really interesting thought. I didn't about the specificity. I don't know if you know anything on specificity of working memory and obesity about food, but
that was just people who with obesity and without.
You had a question.
Oh yeah, so my question was the liking and wanting to use data was the brand.
Yes.
And so and then the image. So yeah, the understanding whether basically so the liking and wanting was like a sip and spit to the taste itself or to the logo, and then the brain imaging was to the logo, and then have taste in the scanner. And really thinking about, because like for me, I think about those as multiple cues, right? Where you have the taste being paired with the sweet oral sweet taste, as well as opposed to just consequences of sucrose being then assigned to taste, and then bringing that to the visual.
Yeah, we didn't feed, we had we did have, okay, so backing up, yeah, no, you're good. The liking and wanting, we didn't see, there was a right DLPFC liking effect to when they're consuming the taste.
So rated liking of the taste correlated
With right DLPFC.
response to it.
We didn't see anything as far as effective intervention to the taste.
Really?
Not anything above threshold. I can say that, I mean, we're talking about randomized at PFWE at 0.07. You have sugar resulting in increased striatal response, mid-insula AIFO, and oral somatosensory to both beverages combined. Not sugar specifically, but the sugar resulted in the sugar condition. Exactly. And let me think. I think that was your question.
If you did have taste.
Oh, we did have taste, we just didn't have enough facts. Yeah.
So I'm trying to think about how you could separate out these sort of systemic metabolic issues with generalized behavioral issues. So if you were to stratify, you know, it would be perfect is if you could take individuals with several genetic neurodevelopmental disorders that have extreme obesity that come with, but some of them have extreme intellectual disability, so it might be hard to do it. But if you just could identify families where there is consistent over multiple generations, higher BMI than those without, and then begin to compare, okay, how do they sensitize? Because the question is whether or not there's a metabolic dimension of this that just drives food intake subconsciously and then gets reinforced by some of these other, versus a neural circuit disruption that is, you know, prepotent, and you could have perfectly nice enteric nervous systems and immune systems and all the other metabolic doodads. But so what do you think?
Well, so we did in my group, and then also when Eric, we did do cross-sectional analyses of adolescents that were of healthy weight, comparing those with obese or overweight parents versus non as a risk marker for obesity. And what you would see is you would see an increased striatal response to the taste in the high risk for obesity group. No changes to the Q. We replicated that in a separate sample. So that's nice. So that effect is there. And then my doc student actually showed that the higher risk sensitized quicker as well.
And if you tried with a non-food stimulus that would be considered appetitive and other things.
So that becomes tricky. Because the nice thing about taste, it's a primary reinforcer, right? And what else can you do is reward the money and the magnet. Oh, you won $5, you won 6 points. That's a secondary reinforcer. You're getting money to be in the study anyway. Are you going to go buy food with it? But we have done some just like monetary kind of, not even really a monetary incentive delay task, but just exposure to winning money and that kind of thing in our group. And what seems to differentiate it is when you're just looking at raw, like, I don't know about the sensitization on that. I don't know that at this point. We are running a behavioral test that would answer that. But yeah, it just becomes because they become so qualitatively different when you're talking about reward. And I mean, we're talking about like, what is a reward? But yeah, there is specificity there. I'm just not sure where.
We've got a couple of questions from our online audience. Quickly, one's from one of our graduate students says, you ever look at how texture affects people's choices and preferences?
Oh, no, but we should. No, we should, especially when we're talking about kind of oral somatosensory regions and those kind of things. One of the things that I do. Sorry?
Milkshakes, but they're thicker.
Well, yeah, so we did, and well, this is interesting. We did Yeah, we did very macronutrient content in milkshake, sugar and fat across the board, which we did not control adequately for texture. And I'm surprised that didn't come up in a review. But yeah, it's interesting because I'm typically because I'm trying to thin things out and just focus on sugar at the moment. So I opted not to do the milkshake anymore and just do more of kind of like, just Kool-Aid and Coke and stuff like that. But yeah.
The other question here is, can you tell us how your findings can be applied to practice, communities, policies?
Yes. That's a good one. When I wrote this grant, reviewer 3 said, this is a really nice study. It doesn't, even if you're right, it's not going to change how we think about sugar. And they're right. Sugar's not great. But you knew that coming in. I'm sorry, I don't give you any more take-home messages than that. No, but when you think in terms of how this is set up, I have a bunch of colleagues that do, the easy go-to there is a sugar-sweetened beverage tax as far as informing policy of saying, hey, we always typically think of sugar-sweetened beverages causing dental caries or increased weight, type 2 diabetes, those kind of things. Well, it's like, no, these things are happening on 18 exposures. And you talk about brand susceptibility. I have a lot of friends that do warning label work or front of package labeling or like removing all package labeling. That do international work in like Peru, where those interventions are effective as far as reducing like sugary cereal intake when it doesn't have toucan Sam on the front.
When we see this research on obesity, we tend to then think of it as a homogeneous condition, but there's a lot of interest in it, whether there's identifiable subgroups and how they might be different. If you have any insights about that.
Oh, as far as here, one of the ways we've looked at that is, and Dana and Alex and I have all at one point in our careers looked at the TACA1 allele, which is associated with compromised dopaminergic functioning as a model to kind of parse out individual differences. We do sweet likers, which is non-sweet likers. We'll do a lot of those kind of tests to see if there's individual differences there. Right now, we're getting into more of we're just dipping our toes into sweet taste receptor kind of variability, but beyond that, we do look at a number of things, but we're not really targeted on a specific individual difference factor. I would have to say mostly like baseline brain functioning, like maybe neuromelanin scans, the dopaminergic regions of NTS and that kind of thing, just to look at propensity of that. But my big exciting thing now is if you're familiar with animal work and reinforcement learning, those kind of things is sign tracking and goal tracking, where you have these mouse phenotypes that behave differently after Pavlovian conditioning. One of them, the goal trackers are one's at higher risk of overconsumption, staying on cocaine, all these things, and we haven't been able to really apply that to humans. And I think we can do that as kind of a almost implicit behavioral phenotype to parse out. But, there's a wealth of info, and with all these big data sets coming out and imaging, it's really nice to be able to explore some of those questions that you wouldn't be able to otherwise.
Yeah. So along the same lines, one potentially important individual difference, behavioral level, the intention to lose weight. Did you measure?
Oh yeah, restraint. Yeah, dietary restraint. We had a number of like the kind of the food cravings and dietary, but we did ask that they had not come off. They've been weight stable for like 3 months, and we measured dietary restraint. No differences between the two groups here. I have done some early work with dietary restraint correlating with DLPFC activity to food pictures and anticipated taste on the way. But yeah, I haven't messed with that in this data set here, but yeah.
Gonna go in the opposite, like from the weight gain. And again, I'm from animal field, so I'm really apologize if I was clicking on familiar with literature. But to gain it, you give the rodent food or like fat, they eat, don't gain weight. You give sugar, gain weight, but not so much. The combination of both, like shake, something like this. This is where you get like 10-fold higher food consumptions. how does that work from the reward? Is that reward more?
I think it's a combination. I mean, and Alex probably, you know, this might be more your, so yell at me when I'm wrong. I've always kind of speculated, and the big speculation there is like, you know, sugar is invoking a reward-related response, or a lot of the reward-related response, but calories more energy, fat is more energy dense, right? You're born with an innate preference for sweet. You're paired that with fat really early on through breast milk. So I think, I mean, it's really hard to parse out in humans though, right? So, I mean, if you could be my reviewer for my next grant where we're going to parametrically do sugar and fat and then sugar and fat, I love that. But yeah, I think, and to speculate, I was always in my head, I was like, well, sugar is going to kind of get you in, and then fat's what's really packing in the calories a little more. But that, like I said, that's just me off the cuff. Yeah.
So I'm wondering if you did the mouse experiments like leptin mutants or Fralin mutants or the mutants that both due to hypothalamic circuitry and also metabolic regulation in the brain gain enormous amounts of weight.
Yeah.
Whether or not the behaviors would be stable, but it's just their metabolic state, or whether or not the behaviors go with it.
Yeah. I'm trying to think back to a talk chat I was having with Lex Kravitz, but yeah, I think it's, you know, I know the handful of kind of the leptin deficient fMRI food studies and those kind of things, but those were small samples incredible studies, but small samples a while ago, and they didn't really assess behavior. But I mean, that would be,
yeah,
that would be where we're at. I mean, we're messing around with dopamine, like D2 agonists with this kind of stuff and those kind of things, but nothing. I wish we just need human opto. Yeah. Actually, they have it now. I think I saw one. Anyway. Oh yeah. All right. Anyone else? All right.